Thank you, everyone. I'm very grateful to be here to speak on behalf of, Biogen and myself, and some of the work that we're doing at Biogen using AI and ML techniques. I was reflecting in the previous talk.
So welcome in the beautiful Ravenna in Italy. Today we will be reflecting on the topic of how great data and artificial intelligence can reveal progression in MS faster.
During my PhD, I was very happy that I was able to recruit 24 patients for my
We
study.
believe that in the world of technology and in the world of medicine, the people in those two worlds speak two very different languages, and we aim to bring those people together. We will build bridges across the different sessions and reflect on this key topic.
We'll start the presentation by giving you a bit of a background to Biogen Digital Health, which is the organisation.
In the closing panel debate, we will reflect in a multi-stakeholder setting on what the future recommendations would be and how together we can actually reveal progression in MS.
So about a year and a half ago, Biogen Digital Health was formed. A new unit at Biogen with the vision of transforming Biogen and patients' lives by making digital and personalised medicine in neuroscience a reality. The organisation was formed from three disparate parts of the organisation in the R&D, commercial and medical parts of the organisation. As I've already mentioned, I think this was in recognition of the fact that the field is moving forward, and the ability to leverage big data sets is something that industry, there's an onus or responsibility on industry to take this seriously, given the wealth of data that we have available to us. So the Biogen Digital Health Digital Health Organisation really has three principal areas of focus. The first is to develop personalised medicine capabilities. The second is to improve the customer and patient journey through digital, clinical, and patient solutions, and to explore new business opportunities such as digital interventions. My group itself is the Personalised Medicine group. It was previously called Reverse Translation, and really, as I've already mentioned, the purpose of my group is to drive new insights in the disease areas that we work in, mainly MS, but also Alzheimer's and Parkinson's, using the wealth of data that we have in-house at Biogen. So here I wanted to kind of start the talk with some work that we've been doing in our group, which I think has direct relevance to progressive MS. So since the 1990s, we all know that MRI has been readily used in MS in clinical trials and in clinical practice for diagnosis, monitoring patients, and providing some kind of prognostic information. But we know that each MRI scan has over 1 million voxels of information that can be extracted, and the current the norm is for neuroradiologists to produce a summary report, which is helpful, as I said, for diagnosis and for measuring disease activity. But I think we increasingly of the recognition that there's a huge amount of information that we may not be able to capture with our current techniques from these images. So here you can see some of the examples of what we now can do with the MRI scans that we have. We're able to segment lesions, obviously, but then we're able to also extract features at a voxel level using things like radiomics, and then apply machine learning and artificial intelligence approaches to develop prognostic or predictive models. And what I wanted to do today is give you some examples of some of the work that we're doing, and also to emphasize the fact that the current way that we interpret the MRI scans may be missing a huge amount of information that's not apparent or visible to the human eye. So on this slide you can see what we capture in our clinical trials. These are 4 very large clinical trials, not just from Biogen, but this identifies or shows that the current ground truth disease activity that we capture in our clinical trials probably substantially underestimates what is really going on at a pathological level. So here you can see on the right-hand side that the average number of individuals with at least one gadolinium lesion at baseline ranges from between about 23% and 40%. But if you look at the equivalent age groups on pathology studies, that percentage goes up to about 70 to 90%. Based on this, we really believe that there's a huge amount of information that is invisible to the human eye, but may be captured using machine learning and AI techniques, and may be helpful for disease prognostication and development of novel biomarkers for patients with MS. So here are some definitions of artificial intelligence. I think the one that is probably most relevant to the work that we're doing is the ability to find hidden or latent relations between data that correlate or have a causal relationship with outcomes that are invisible to the human eye. Here you can see a MRI scan from an individual who has no evidence of active disease, so no gadolinium-enhancing lesions and no new T2 lesions. And you can see on the T1, the T2 and the FLAIR images that there is substantial amounts of signal heterogeneity. And the hypothesis that we were working with is that this difference in signal intensity may underlie some differences on a pathological level, so in terms of axons, remyelination, demyelination, and immune cell infiltrates, microglia, macrophages, lymphocytes. And that was really what we have been setting out to do, largely using machine learning AI approaches and our clinical trial data sets. And that's to see whether or not from a cross-sectional pair of T1 and T2 scans, you're able to train a machine learning classifier that can detect at a voxel level areas or regions of active or chronic disease at that single cross-sectional time point without the need for gadolinium or a reference scan. So i.e. trying to identify temporal dynamics on that single cross-sectional scan. You can see on this image here another example where there is some ground truth acute disease activity, and you can see that the models that we are developing are able to detect at a voxel level areas that are indicative of active disease. Actually, this is an individual with no active disease burden, and you can see that the algorithms that we're developing are picking up areas of acute foci at a voxel level. On this next slide, you can see a patient who does have some ground truth active disease activity, and again, you can see quite nicely that the algorithms that we developed at that single time point are able to detect at a voxel level areas of high probability of acute disease activity. And obviously, this kind of approach can also be used to, for example, detect chronic active disease that may be more relevant to the progressive biology in MS, and I'll talk about that on the next slide. So, there are obvious use cases for the work that we're doing in terms of optimizing the way that patients are managed in clinic, where, for example, a gadolinium scan may not be available or a reference scan isn't available, but there are also direct applications of these types of work to capture other aspects of MS disease using machine learning AI. So here on the left-hand side, you can see some work that we presented at a conference this year where we were able to with modest accuracy, discriminate slowly expanding lesions from non-slowly expanding lesions at a single time point. And similarly, on the right-hand side, we were able to train algorithms that can identify areas in the normal appearing white matter that are at risk of future lesion formation, which in and of itself may represent a new treatment paradigm in MS that we could test with preventative therapies, for example. So ultimately, I think the objective of utilising the huge amounts of data that we have in-house is really to develop algorithms that may be able to provide better care for patients and to better understand their disease. So using the MRIs, we really strive, our ambition is to be able to extract as much information as possible using radiomics, machine learning and AI to develop ways to automatically detect different types of MS lesions with much higher sensitivity than the ways that we currently do this. I think this has already been alluded to, but another project that we're working on is to use similar input information and leverage ML/AI techniques to identify individuals who are most likely to respond to a specific therapy, taking into account what their individual prognosis may be with and without that therapy. So why do we at Biogen want to do this? I think, as I've already alluded to, we have huge amounts of data. I think we have a responsibility, given that data, to derive these new insights that may be helpful in a clinical setting. But there are obvious use cases as well from an R&D perspective, not least to identify patients who may be most likely to demonstrate proof of biology or proof of concept in our clinical trials, so as a way of enriching a population for patients, for example, who have high evidence of chronic active disease in the way that we were able to show on that earlier slide with the cell, non-cell lesion classifier. But also, through this approach, we identify radiomic features or biomarkers that may also serve as pharmacodynamic markers in our clinical trials, so that we can actually establish proof of biology at the earlier stages and increase the probability of success of our trials. So I'd like to thank everyone for their time and looking forward to the discussion.